Good day, everyone. My name is Dr. Paul. I am with the School of Healthy Aging, Medical Aesthetic and Regenerative Medicine, UCSI University. I would like to welcome you all to this online video CME. In this video, I'm going to talk about the physiology of aging in pregnancy. This is the overview of this CME. We will discuss what are the physiological and cellular characteristics exhibited during the pregnancy and aging, and also comparing the common biomarkers, pathologies, and genetic and epigenetic effects between aging and pregnancy. What are the natural antioxidants and how to optimize a healthy aging during the pre and postpartum. First, what do you know about aging? Aging is easy to recognize but difficult to define. There are many of definitions out there, and most of the definitions of aging indicate that it is a physiological process associated with decline and deterioration of functional properties at the cellular, tissues, and organ level. The accumulations of biological changes over time leading to decreased biological functions and impaired ability to adapt to stressor. There are 5 types of aging: chronology aging, psychology aging, social aging, cognitive aging, and biology aging. In this topic we discuss is more on the biological aging. What is biology aging? This aging is also known as physiology aging. Biological aging is the gradual deterioration of function characteristic. It refers to the physical changes that slow us down as humans get into middle and older years. For example, artery might clog up, or problems with the lungs might make it more difficult for us to breathe. Body experiences changes in functions during periods of metabolic stress. This process is manifested during the aging and pregnancy. There are parallels both in development of pathology in aging and pregnancy. However, aging is characterized by a linear degradation without recovery, whereas in pregnancy, there are potential recovery that reverse the damage caused by the stress to original level postpartum. In this section of inflammation and signaling, we will look into the oxidative stress and antioxidant activity, markers and mediators of inflammation, insulin-like growth factor 1, and autophagy in pregnancy and aging. Oxidative stress and antioxidant activity. What is oxidative stress? Oxidative stress can occur when there is an imbalance of free radicals and antioxidants in the body. It occurs when reactive oxygen species accumulate excessively beyond the scavenging activity of antioxidants. The body cells produce free radicals during normal metabolic processes. However, cells also produce antioxidants that neutralize these free radicals. In general, the body is able to maintain a balance between antioxidants and free radicals, and the balance between pro-oxidants and oxidants is tightly regulated and extremely important for maintaining vital cellular and biological functions. Several factors contribute to oxidative stress and assist free radicals production. These factors can include diet, lifestyle, environmental factors such as pollution and radiations. The body's natural immune response can also trigger oxidative stress temporarily. This type of oxidative stress causes mild inflammations that goes away after the immune system fights off infections or repair an injury. Chronic overproduction of oxidants activates the cellular pathway. Stimulations of cell proliferation damage to cellular protein, DNA, and lipids. Uncontrolled oxidative stress can accelerate the aging process and may contribute to the development of various chronic diseases. What are free radicals? Free radicals are also known as pro-oxidants. There are any chemical species capable of independent existence with one or more unpaired electrons. Free radicals are highly unstable and reactive. They're looking for electrons from other sources to stabilize themselves. In this process, they initiate a chain reaction of oxidation. Most common free radicals are reactive oxygen species. Reactive oxygen species, they can be oxygen-derived radicals or oxygen-derived non-radical species. They are now considered as a major player in biochemical reactions, cellular response, and clinical outcome, and also involved in normal aging processes as well as in aging pathologies. This is a study of oxidative stress in healthy pregnancy and preeclampsia. They conclude that stable oxidative stress level maintained throughout the healthy pregnancy, demonstrating the balance of oxidative stress, and is curing by antioxidant pathway to maintain the healthy function. While in preeclampsia pregnancy, systemic inflammations and superoxide concentrations are higher and result in a deterioration of microvascular endothelial functions. These findings support the hypothesis that vascular functions is directly linked to the amount of oxidative stress, suggesting an important place for antioxidant treatments in the preventions on preeclampsia. Oxidative stress has been implicated in the pathogenesis of many pregnancy disorders, such as preeclampsia, hypertension, and spontaneous abortions. As mentioned in previous slide, free radicals can damage all cellular macromolecules, including the proteins, lipids, and nucleic acid. This destructive effect play a role in the onset and progression of different diseases and in normal aging. Lipids are highly prone to get oxidized. Free radicals disrupt the phospholipids in the cell membrane to form lipid peroxide, a process known as lipid peroxidation. Malondialdehyde, MDA, is an end product of lipid peroxide metabolism, serves as a biomarker for lipid peroxidation determination. This is a finding from a review study which published in year 2020. The oxidative stress and antioxidant activity in pregnancy and aging are summarized in the table in this review article. I will talk about the antioxidant activity after this. This table shows that the oxidative stress pathway that target the MDA in pregnancy raise in pre and eclamptic pregnancy, highest in first trimester, increased level in treated and incomplete abortion, increased level in pregnancy-induced hypertension, and correlations with elevated lipid level. Decrease in second and third trimester inversely correlated with Throx level that increased at the same time. Decrease from pre-delivery to post-delivery in multiparous women. While in aging, it increased with age in cataract patients and increased level in hypertension. Next, antioxidant. Antioxidants are compounds capable of preventing the pro-oxidation process or biological oxidative damage by scavenging or stabilizing the reactive oxidative species. And why are antioxidants important? Because they inhibit the conversion of nitrate to nitrosamine and enhance the immune response. They terminate the free radicals induce cellular damage and functional degenerations. Vitamin E and C, ubiquinone, remove the free radicals from the epidermis of the skin and counteract their potentially damaging effect. And they trap and neutralize the free radicals and protect our body tissues from environmental pollutants. The body encloses a complex antioxidant defense grid that relies on endogenous enzymatic and non-enzymatic antioxidants. Antioxidants enzymes are to defend against the oxidative stress, which remove the reactive oxygen species. The first-line defense antioxidants include superoxide dismutase, catalase, and glutathione peroxidase. In pregnancy, the antioxidant activity pathway that target the catalase increase during the third trimester in correlations with MDA, decreased level in treatment and incomplete abortions, blood level decrease in GTM, 25 to 70% increase in pre-delivery level, while in aging, reduced to 38% of normal in cells lead to senescence. The next antioxidant activity pathway that target the glutathione peroxidase stable throughout the pregnancy. However, in aging, decline with age. The superoxide dismutase increased during third trimester in correlation with MDA. In here, I would like to talk a little bit about the source of natural antioxidant. Most common antioxidants found every day in foods are vitamin C, ascorbic acid, vitamin E, tocopherol, vitamin A, carotenoids, various polyphenols including flavonoid, anthocyanin, a type of flavonoid. So these are the antioxidants we do take regularly in our diet. Vitamin C in most citrus fruit, some vegetables and tomato. Vitamin E in cereal grain, broccoli, sprout, cauliflower cooking oil, almond, black rice. Beta-carotene in vegetables such as spinach, tomato, carrot, sweet potatoes, apricot, papayas. Flavonoids in potato, tomatoes, lettuce, onion, wheat, dark chocolate, grapes, red wine, black tea. Various polyphenols in green tea, many red or purple colored fruits or vegetables such as the red cabbage, blueberry. Lycopenes in tomatoes, papaya, watermelons, pink grapefruit or guava. CoQ10 can be found in wheat bran, fish, chicken liver. Vitamin C is a naturally occurring organic compound, act as a powerful antioxidant which help to scavenge the free radicals. It is also believed that vitamin C helps to delay or even prevent age-related cataract and macular degeneration of the eyes. Vitamin C may also play a role in preventing atherosclerosis. Study found that people with atherosclerosis has low level of vitamin C in their bloodstream, implying that these people were not getting enough of vitamin in their diet. Vitamin E, tocopherol, its major function appears to be a non-specific change-breaking antioxidant that prevent the propagation of free radical reactions. It has been studied extensively in mammalian research as membrane stabilizer and multiphase antioxidants that scavenge oxygen-free radicals, lipid proxy radicals, and single oxygens. Vitamin C combined with vitamin E to regenerate the alpha tocopherol from alpha tocopherol radicals in the membranes and lipoproteins, and also raise intracellular glutathione levels. Coenzyme Q10 is a vitamin-like substance found throughout the body, but especially in the liver, heart, kidney, and pancreas. It requires for the proper functions of many organs and chemical reactions in the body. It helps to provide energy to the cells. Study reported that a higher level of plasma CoQ10 was significantly associated with reducing the risk of coronary artery disease. Their results support that potential cardioprotective impact of coenzyme Q10. Another study showed that supplementations with CoQ10 in infertile men can reduce oxidative stress in seminal plasma and improve the semen parameter and antioxidant enzyme activity. Studies have shown that CoQ10 levels are low during inflammatory conditions. CoQ10 supplementation decreases the C-reactive protein and other inflammatory mediators. Okay, we come back to the section of inflammation and signaling. Just now we talked about the oxidative stress and antioxidant activity. We continue with inflammation. The following are the causes of inflammation in our body: trauma, microtrauma, intense physical activity, chemical, infections, increased body fat, nutritional imbalance. A study concluded that chronic low level of inflammation contribute to the pathogenesis. And chronic inflammations can affect on digestive stress, chronic pain, lower immunity, fatigue, nervous and hormonal imbalance. Human aging is characterized by a chronic low level of inflammations, and this phenomenon has been termed as inflammaging. Inflammaging is a highly significant risk factor for both morbidity and mortality in the elderly people. Chronic inflammations to be a major factor in the development of degenerative disease and loss of youngful functions. This is triggered by cellular stress and dysfunctions, such as they are caused by excessive calorie consumption, elevated blood sugar level, and oxidative stress. Higher level of inflammatory markers correlated with an increased level of adipose tissue, decline of sex hormones, and an increase in oxidative stress. The markers and mediators of inflammation are tumor necrosis factor alpha, C-reactive protein, nuclear factor kappa B, and interleukin. Tumor necrosis factor alpha is a cytokine released during the cellular damage, stress, or infections. C-reactive proteins is a protein rapidly produced by the liver during an inflammatory response. It caught the damaged cells to make them easier to recognize by other immune cells. It raised in cerebral cancer, rheumatology, gastrointestinal, and cardiovascular conditions and infections. High sensitivity C-reactive proteins is strongly associated with elevated risk of cardiovascular disease and stroke. Nuclear factor kappa B, it turned on the expressions of over 400 genes involved in the inflammatory response cytokine and pro-inflammatory enzyme including the cyclooxygenase 2 and lipoxygenase. The nuclear factor kappa B pathway is a noted mediator of aging, driven in response to a variety of stress, including oxidative stress and genetic damage. Interleukin, a group of cytokine, promotion and resolution of inflammations. The pro-inflammatory interleukins such as interleukin 1 beta, interleukin 6, and interleukin 8. In this review, reported that in pregnancy, tumor necrosis factor alpha were not detected change in level between women with recurrent pregnancy loss and parous women. Polymorphism in promoter regions found associated with recurrent pregnancy loss in a meta-analysis. Higher concentration in women at risk of premature pregnancy terminations compared to women with healthy pregnancy, and higher level in preeclamptic pregnancy. While in aging, tumor necrosis factor alpha is predictor of mortality. It increased by almost twofold in centenarian versus a young control. High plasma concentrations associated with dementia, Alzheimer, and atherosclerosis. Higher level associated with high triglyceride and low HDL total cholesterol ratio. Continuous increase in type 2 DM patients and inversely related to the muscle strength and mass in geriatric patients. C-reactive protein is reported high level associated with preeclampsia, and high level of C-reactive protein is reported associated with hypertensive cardiovascular disease in aging. Nuclear factor kappa B in pregnancy, downregulated in pregnancy, leading to a shift from T helper 1 to regulatory T cells promoting allograft tolerance to avoid the immune response against the fetus. Downregulated in pregnancy, leading to a shift from T helper 1 to T helper 2 for anti-inflammatory response for healthy pregnancy maintenance. Elevated in peripheral blood mononuclear cells of pre-eclamptic women were reported in another study. While in aging, upregulations of nuclear factor kappa B is reported associated with Alzheimer's disease, Parkinson's disease, type 2 DM, atherosclerosis, osteoarthritis, osteoporosis, and overactivity lead to more rapid onset of cognitive decline. Insulin-like growth factor 1. Insulin-like growth factor 1, also known as somatomedin C, is a 70 amino acid protein. About 70% is produced by the liver in response to human growth hormone, which is produced in the pituitary gland. Insulin-like growth factor 1 is responsible for a wide range of cellular functions and is required for normal growth and health maintenance. These functions include cellular differentiation, transformations, and apoptosis suppressions. Clinical evidence show that low insulin-like growth factor 1 level play a key role in the degenerative changes brought about by the aging process. Insulin-like growth factor 1 signaling has been shown to influence mammalian aging and related pathogenesis. This study showed that females with insulin-like growth factor 1 level below the medians had significantly longer survival compared with females with level above the medians. They conclude that low insulin-like growth factor 1 level predict life expectancy in exceptionally long-lived individuals. In this article, it shows a significant fall in insulin-like growth factor 1 during the first and second trimester, followed by an increase. The initial reductions during the first trimester has been hypothesized in another article to be due to the liver resistance to the pituitary growth hormones due to the estradiol, secretions of which inhibit the interactions of growth hormones on JAK2 phosphorylations, decreasing the insulin-like growth factor 1 synthesis. Autophagy. What is autophagy? To keep it simple, autophagy is the housekeeping of the cells. In terms of former, autophagy is an internal process that helps in the lysosomal degradations and the removal of old and unwanted cellular molecules, including the protein, ribosome, lipid droplets, and other organelles. It is how our body recycles damaged protein and organelles. Autophagy is important because when your cells undergo autophagy, they take a break from dividing, and that's a good thing. You don't want your cells always dividing because that will create free radicals. We have already discussed in previous sections of oxidative stress. Too many free radicals are associated with cellular damage and a wide range of disease. Autophagy is important throughout our entire life, but it is especially critical as we age. The cell capability to degrade its organelles and perform autophagy tend to decline with age, predisposing unnecessary elements such as protein and the debris start to build up, which may become harmful. Autophagy is critical in management of free radicals, aiding as a mechanism to turn over damaged proteins and organelles such as mitochondria. Studies have linked to build-up of proteins to disease like Alzheimer's and Parkinson, as well as other memories and movement disorder. Another autophagy-related issues that a lot of people have is osteoarthritis. Studies reported that autophagy-related genes were reduced in human chondrocytes from patients with osteoarthritis. This study reported that the upregulations of autophagy may play a role in the pathogenesis of fetal growth restriction. Another study showed that multiplarity is associated with decreased level of autophagy in peripheral blood mononuclear cells. Dysregulated autophagy might be one mechanism leading to the spontaneous abortions in nulliparous women. Next, let's discuss the physiological pathologies between pregnancy and aging. Physiological pathologies often share symptoms and risk factors with aging and pregnancy. We will look into the cardiovascular pathology and diabetes in these physiological pathologies. Cardiovascular pathology. So what happened in the cardiovascular pathology between pregnancy and aging? Aging is the most significant risk factor for cardiovascular disease. Aging effect on the cardiovascular system often lead to the complications such as atherosclerosis, hypertension, and thrombosis. Surveys show that 70% of older adults suffer from hypertension. A similar phenomenon is found in pregnant women at the start of gestation. So it is important to understand that even in normal patients, pregnancy imposes some dramatic physiologic changes upon the cardiovascular system. The stress of pregnancy causes a 17% and 15% increase in pulse and cardiac output respectively. Hypertension is the most common medical disorder in pregnancy. Various types of hypertension affecting pregnancy are pre-existing hypertension, pregnancy-induced hypertension, and preeclampsia. Pre-existing hypertension affects 1 to 5% of pregnancy. pregnancy-induced hypertension to occur in 5 to 10% of all pregnancy, where gestational hypertension can be as common as 25% in the first pregnancy. The incidence of preeclampsia has increased, possibly due to an increased prevalence of predisposing factors such as chronic hypertension, obesity, and diabetes. Hypertension disorder of pregnancy, in particular preeclampsia, commonly complicate pregnancy and can even have long-term consequences. Women with early onset of preeclampsia are at an increased risk for future cardiovascular event. Because the risk of developing chronic hypertension could be more than 20%, blood pressure should be checked regularly, and the first focus should be on lifestyle modification. Acute myocardial infarction. Both the aging and pregnant populations show greater risk for myocardial infarction. Although myocardial infarction is a rare event in women of reproductive age, pregnancy increases the risk due to the number of factors. Hypertension is also strongly associated with myocardial infarctions in pregnancy, as it possibly due to the damaged blood vessels that have already undergone changes due to the hemodynamic stress of pregnancy or endothelial activation. At one's age, hypertension and smoking emerge as the most important risk factor. Study reported that the odds of acute myocardial infarctions were 34 higher for women aged 40 years and older than for the women aged 20. Hypertension increased the risk 20-fold and smoking 8-fold. Type 2 diabetes mellitus and gestational diabetes. Type 2 DM is one of the most common diseases in the elderly. It shares many features with GDM. Both are common metabolic disorders manifesting glucose intolerance and insulin resistance. Moreover, the mechanism for development of either type of diabetes in pregnant and the elderly are very similar. Studies show that they are linked to both oxidative stress and inflammation, as well as common causes such as beta cells dysfunction. During the gestational period, GDM may increase the likelihood of complications, such as abnormal blood glucose, fetal hyperinsulinism, macrosomia, or stillbirth. Study demonstrated in a meta-analysis that women with GDM have a greater risk of developing type 2 DM. Lifestyle risk factors such as diet high in refined sugars or lack of exercise exacerbate the progress of GDM to type 2 DM. Modifying this lifestyle approach can lead to an improvement in the condition. Genomic modifications and genomics. So here, we'll talk about the telomeres, DNA methylations, and epigenetic and genomic. Telomeres. Telomeres protect the vital information in our DNA. They are located at the end of each strand of DNA like a cap that protects our chromosomes. They are an essential part of human cells that affect how our cells age. As a normal cellular process, a small portion of telomeric DNA is lost with each cell division. When telomere lengths reach a critical limit, the cell undergoes senescence or apoptosis. Telomere shortening is involved in all aspects of the aging process. On a cellular level, it represents our biological age. Many scientific studies have shown a strong connection between the short telomeres and cellular aging, but telomeres can also be shortened by stress, smoking, obesity, lack of exercise, and a poor diet. Progressive telomere shortening with aging was studied in the normal liver tissue, and it reported that telomere length demonstrated age-related shortening with reduction of 55 base pair per year. A cross-sectional study was carried out involving 1,954 women from US to examine the association between the parity and the leukocyte telomere length. The findings suggest that parity may associate cellular aging. The geometric mean telomere length was shorter in women who have been pregnant compared to non-reproductive women. Another study involving 19 mothers with healthy child as a control group and 39 mothers with chronically ill child as a caregiving group was carried out. Mean telomere length and telomerase activity were measured. The average perceived stress level was higher in the caregiving than in control. This study showed that the high stress group has shorter telomere and lower telomerase activity. Therefore, they conclude that stress is associated with indicator of accelerated cellular aging. This study reported that those with more surviving children were associated with having greater telomere length and evident that multiparous women had longer telomeres. This protective effect may be explained by the actions of the gonadal steroid estradiol, which increased dramatically during pregnancy, up to 200 times non-pregnancy level, and is known to act as a telomere length protective factor. Indeed, estradiol is a potent antioxidant that reduces the cellular exposure oxidative stress. Oxidative stress is known to lead to the telomere length shortening. Women who experience more pregnancy are exposed to higher estradiol level, which indirectly protect the cells from telomere length shortening by reducing the exposure to oxidative stress. Moreover, the estradiol also increases the telomerase activity. In this study, it showed that women with late puberty, 34 to 37 years old, has increased the odds of being in the longest tertile of telomere length compared with the women who have their last child by age 29. They conclude a tenuous genetic link between the reproductive lifespan, longevity, and mechanism of aging. However, future studies to understand the base of this association are required. Telomere length varies greatly throughout the life and throughout body tissues and cell type. The mean telomere length of leukocyte is widely accepted as a biomarker of age and health status. Future studies needed to understanding the mechanism by which postpartum telomere length is maintained and restored could be possibly lead to the treatment and better maintenance of telomere length in the aging population. DNA methylation and epigenetics. DNA methylation is one mechanism of controlling DNA replication. During the methylation, a methyl group CH3 is added to the base. When that happens, it changes the structures of DNA. It usually occurs in the CpG islands. The CpG islands locations they are heavy in cytosine guanine base pairing. And this is how the DNA methylation look like. Let's talk about the DNA methylation a little bit more. DNA methylation is essentially inactivating the DNA transcription. So in this figure, the DNA methylated at the CpG islands, the black dot, and the histone are deacetylated. The gene cannot be transcribed. This is one mechanism of epigenetics. And epigenetic is how our environment and all the things we interact with affect our genetic. Not just our gene themselves, but everything else affect our gene transcription. DNA methylation is a stable epigenetic mark. It's one of the strongest markers of aging, with current research characterizing an epigenetic clock of aging based on DNA methylation. Studies reported that aging and associated pathology are often characterized by changes in methylation level, specifically the genome-wide hypomethylation and the CpG-specific hypermethylation. Methylation of target site on the genome is maintained by the DNA methyltransferase and removed by the demethylase. This study compared the methylation changes in the CpG island in the T helper 1, 2, and 17, and the regulatory T cells pathway gene to identify the epigenetic effect on the cytokine shift to the T helper 2 response. Following a cohort of 54 women from the age of 18 through to the full term of pregnancy, They found that the T helper 2 pathway methylation changed in the first and second half of the pregnancy, and the T helper 1 methylation changed in the second half of pregnancy. In the case of T helper 1, 27.7% of CpG in the first and 36.1% in the second half were significantly differentially methylated compared with the control, and 35.1% and 33.8% in T helper 2 respectively. Genomics. First, what is genomics? Genomics is holistic in scope. It studies all the genetic information in an organism and how the genome interacts with its environment. Genomics is high throughput. It reads and analyzes large amounts of DNA information quickly and efficiently. Genomics is resource generating. These resources further enable study and discovery. The availability and cost effectiveness of technologies such as whole exome and whole genome sequencing have enabled the development of large dataset analysis and statistical tests, such as the Genome-Wide Association study. Such studies are widely conducted in attempt to define the genetic variation that is associated with polygenic and complex phenotype, such as aging and longevity. This study is a whole exome sequencing performed on blood sample of pre-eclamptic women. In this study, the 50 pre-eclamptic women surveyed were found to have reduced genetic variation in comparison with the 50 healthy pregnancy control, suggesting a protective role of genetic diversity against the development of preeclampsia. Whole exome and whole genome sequencing can allow for identification of genetic variation association with phenotype of pregnancy and aging, to identify relevance of single nucleotide polymorphism. Unfortunately, there is a little data concerning the genomic of pregnancy. Most of the available studies focus on the outcome in the newborn offspring and less on the recovery mother. In order to truly understand the genetic and epigenetic mechanism of the recovery and regeneration of the postpartum woman, tightly controlled and phenotypically characterized genome-wide association study need to be conducted. Lastly, how do we optimize healthy aging during pregnancy and after delivery? Although everyone knows the benefit of maintaining the physical exercise, but when it comes to the pregnancy exercise, most mothers-to-be have reasonable safety concern. Except for women with non-contraindications, studies have shown that exercise for pregnant women is not only safe, but also beneficial for their babies and their own health during the pregnancy and after birth. Studies have found that the benefits of exercise during pregnancy not only help the woman endure the demands of childbearing, but also reduce the risk of pregnancy complications like gestational diabetes or preeclampsia. Ease the pregnancy discomforts like back pain or constipation, promote a healthy and steady weight gain during pregnancy, reduce the stress and anxiety and improve sleep, strengthen the heart and improve the circulation, prepare the body for labor and delivery, allow for faster recovery after birth. In pregnancy, the center of gravity changes. Extra strain are placed on the joint, muscle, and lower back. Therefore, the exercise like Kegel, swimming, prenatal yoga are the recommended exercise during pregnancy. The exercises to avoid are those that involve heavy lifting or standing for a prolonged period of time, increase the risk of falling or abdominal trauma. Postnatal exercise is important because it can strengthen the pelvic floor and tummy muscle, improve the quality and quantity of sleep, reduce the body weight, depression, anxiety, and improve the psychological well-being, reduce the risk of developing heart disease, diabetes, and cancer, increase metabolism, reduce high blood pressure, high cholesterol, and prevent back pain. When can start exercise after delivery? If uncomplicated natural birth, can resume exercise as soon as the mother feels comfortable enough to do so. C-sections or complicated natural birth, it recommended waiting 6 weeks or after the postnatal examinations. These recommended postnatal exercise are listed here. The pelvis floor muscle exercise, the Kegel exercise. Pelvis floor muscles are stretched during the pregnancy and childbirth. Strong pelvis floor muscles are essential to prevent the leakage of urine when sneeze or cough, to give complete control of bowel and to prevent the UV prolapse. Other exercises like abdominal bracing exercise, brisk walking, yoga, low-impact aerobic workout, lightweight training, and bicycling are recommended. Spirituality and relaxation is important in release the stress. Stress is a physical, mental, or emotional factor that causes the bodily or mental tensions. Stress happens when the body could not cope with pressure, which triggers the psychological responses. Therefore, it is important to get involved in regular periods of personal relaxations. Setting aside time for yoga, meditations, deep, slow breathing, relaxed lifestyle. There are several ways of maintaining an internal balance, such as love, religions, meditations, and intuitions. Diet and nutrition. To maintain a healthy pregnancy, approximately 300 extra calories are needed each day. These calories should come from the balanced diet of protein, fruits, vegetables, and whole grains. Sweets and fats should be kept to a minimum. Supplements such as folic acid is important. It helps to reduce the risk of neural tube defect, which are the birth defect of the brain and the spinal cord. These pictures show the example of the healthy snack ideas. For healthy eating in pregnancy, we can refer to this My Pregnancy Plate, created by a clinical dietitian. In My Pregnancy Plate, it recommends us to choose the large portions of variety of non-starchy vegetables such as leafy greens, broccoli, carrots, pepper, or cabbage. Choose a variety of whole fruits, limit juice, and dry fruits. Choose a protein source such as poultry, beans, nuts, low-mercury seafood, egg, tofu, or low-fat cheese. Limit red meat and avoid cold cuts and other processed meat. Choose the whole grains, legumes, and starchy vegetable. Limit white bread, white rice, and fried potato. Choose small amounts of healthy oils for cooking. Aim for at least 30 minutes of walking and another physical activity each day. And choose 2 to 3 servings of non-fat or 1% milk or yogurt. Drink mainly water, decaf tea or coffee, and avoid sugary beverage. It is important to continue to maintain a healthy eating after delivery, postpartum. To maintain a healthy eating, we can refer to a few food pyramids such as a Mediterranean food pyramid, Okinawa food pyramids. And this is the Mediterranean food pyramid. A study showed that adherence to the Mediterranean diet was associated with reduced the risk for stroke and cognitive impairment. Higher adherence to Mediterranean diet was associated with leukocyte telomere length variability and telomerase activity among the elderly people, and also associated with a lower incidence of chronic ill death like hypertension, myocardial infarctions, dementia, vascular diseases, and heart failure. This is the Okinawa diet food pyramid. Okinawa is a small island in Japan. It's well known for their long average life expectancy, high number of centenarians, excellent state of health, and low risk of age-associated disease. Okinawa food pyramid is based on 25 years of research and reflects the eating habit and pattern of longest life, healthiest group of people in the world. This food pyramid is a scientifically proven guidelines for healthy eating. It's based upon the traditional eatery habit of Okinawa elders. They also have a longest life expectancy, uncommon to die from the heart disease, stroke, or cancer. Based on the research, there are four lifestyle factors that explain their longevity: low calories, plant-based diet, regular exercise, strong spiritual commitments, and positive outlook on life. This is the food pyramid specifically designed as an anti-aging diet. Start with 10 to 12 glasses of room temperature pure filtered water a day. Supplement rich to correct the balance of mineral, vitamins, and nutrients, such as high-quality multivitamins and minerals to include the key antioxidants that are missing from everyday diets should be taken. Low glycemic index type fruit should be taken, such as apple or blueberry. Always take whole fruit and avoid fruit juice, which are high in sugars and low in fibers. Low glycemic index foods like whole grains, cereal, barley, and oats. Reduce high glycemic carbohydrate intake, such as rice, wheat, bread, pasta, and rice. 8 to 10 portions of vegetables should be taken. Organic eggs and nuts are excellent source of protein. At least 8 ounces of whole water deep sea fish, such as salmon or tuna, should be taken per week. This will provide the body with the necessary omega-3 fatty acids as well as protein. Red meat, such as beef, should be restricted to once or twice a week, together with desserts such as ice cream and cakes. Thank you.